Gel electrophoresis is a method used to separate DNA fragments. This practice is used for a multitude of things, but I'll be talking about the use for DNA fingerprinting. Here we have an aerogose gel, which is a jello-like slab made from carbohydrates. At one end, there are indents that we call wells. This is where the DNA samples are placed, and here we have two separate samples. We place a negative charge closest to the wells and a positive charge on the far end. The DNA fragments are negative due to their molecular composition, so they gravitate through the gel and towards the positive charge. When looking at the results after a specified period of time, we see a bunch of lines. Each line is called a band, and they represent different fragments of DNA. The pieces that are furthest from the starting point are the smallest fragments, and the largest fragments are the ones closest to the wells. The smaller pieces are able to move farther than the larger ones, due to the porous nature of the gel. Imagine a bear and a squirrel in a dense forest. The squirrel is able to move a larger distance at a faster speed because it can weave through the trunks and branches. A bear is larger and it travels much slower through the same forest. Now, we compare the distances from each DNA sample to each other, and we can see that there are a couple bands that line up. This is how we compare DNA for family trees, crime investigations, and so much more. Now, how are these fragments separated into small and large pieces in the first place? We use a solution that contains a restriction enzyme particular to the type of testing being conducted. The enzyme EcoR1 is most common for gel electrophoresis, and it cuts the DNA strand according to the base pairs. Let's take a closer look. These are the base pairs of a DNA strand. You can see how the enzyme only cuts at certain bases. This is because EcoR1 will only cut the DNA in this particular order. This is a different process for different DNA strands, as each person's DNA is unique to them. While the enzyme will still cut the bases on each strand, it will be in different places. This is why we end up with different sized DNA fragments, which run different lengths in the gel. Let's take a look at how gel electrophoresis is used for DNA testing in criminal cases. Say there was a robbery, and left behind was a broken piggy bank. The police have two viable suspects, but they can't figure out which one did it. Luckily, there is a tiny drop of blood on the broken piggy bank, and we can use that to compare DNA samples. First, we have our sample that was found at the crime scene. Then we have two samples taken from the two key suspects. Once we have used a restriction enzyme to break up the DNA strands, we place them into these wells. When we run the test, we look to see which bands line up best to the sample found. We can see here that the sample matches suspect 2's DNA. This match allows us to clarify who committed the crime. We can also use this practice for paternity testing if there's an unknown father of a child. We take a sample from the child as well as the mother, and then the two suspected fathers. When we run the test, we look to see which bands line up with the fathers. Here we can see that candidate 1's bands line up best, and when we look at the mother's DNA, we can see that her bands line up with the rest of the child's. This is a great example of familial DNA testing and how it works. I hope you learned something new and you learned how cool that biological forensics is.